and I'm a science teacher here at Sequoyah Mid-High High School. Today what I would like to do is do a strawberry DNA extraction lab for you. It's very exciting to me because it's an opportunity to show students DNA in a physical, tangible sense versus we just talk about it. Here's a diagram of it. I'd like to go over a little bit of a background information with you first and kind of make sure you're all caught up to where I'm at. With our DNA extraction lab, the first thing we need to talk about the fact is that the DNA is coming from cells. All living things are made up of cells. It doesn't matter if it's a bacteria or a protist, which are single cells, or if it's multicellular, like a fungus or a plant or an animal. But all living things are made up of cells, and then what's inside of there? It's kind of a layer project. When we look inside of the cells, it depends if you have a eukaryotic cell or a prokaryotic cell. When I talk to students and I explain the difference, the main thing here is a eukaryotic cell is going to have a nucleus inside of it, and inside of the nucleus is where the DNA is. If it's a prokaryotic cell, it still has DNA, it's just not inside of a nucleus. So I always tell my kids, think pro sounds like no, there's no nucleus here. But U actually literally translates to true, and karyote is nucleus. So that goes with it. And so when we look at dissecting down our cells so that we can get the DNA out, we got to figure out, first of all, where's it at and what is it encased in? How are we going to break it apart? Well, we have this cell membrane around the outside that we have to break open, and we also have this nuclear envelope. It's really just a fancy name for a membrane that goes around the nucleus that we also want to break down, and so we're going to work on that today as well. So, a little biochemistry that goes into this cell. When you first look at your cells, there's not just the nucleus and the DNA, there's some other organelles in there, but what makes all that up? It's kind of like saying atoms make up compounds and compounds make up all of the things in our lives. So when we look at the biochemistry of a cell, there's four main parts here in blue. There's carbohydrates, lipids, proteins, and nucleic acids. All of that is going to be inside of our strawberry today. And when we look at carbohydrates, we're talking about sugars. Okay, they can be simple sugars or complex sugars. It depends on how the cell wants to use them. Simple sugars are going to be easier for the cell to quickly use. Complex sugars are kind of how they store it until they turn it into a lipid. If I eat a lot of sugar, it's going to turn into a lipid. We know lipids as fats. So we're going to talk today about some of the things that cells use fats for. Some of it is for storage of energy, but all that membrane that I just talked about a minute ago, it is made up of fats. There's proteins inside of there. We need to break those down as well. Our DNA is actually kind of wrapped around them. And then the word nucleic acid is the second half of what DNA stands for. The Na is nucleic acid. Deoxyribonucleic acid, okay? So it says here, I know it's probably hard for you to see, how are we going to be able to see that DNA? It's so small that you can't even see it when you look underneath a microscope. So how are we going to get that DNA so that it's going to be visible? The first thing we're going to do is break down those membranes so that we can see all of the DNA come out. So there's this layer around the outside of a cell called a phospholipid bilayer. Phospholipid, lipid tells you that it is a fat, okay? And so we're going to break down fats, and that phospholipid bilayer kind of looks like this. There's an outside portion to it that is a phosphate group, and then there's these two fatty acid chains that are attached to it. All of my phosphate groups on the outside love water, okay? And so you'll notice here there's water on the outside of the cell and there's water on the inside of the cell, and all these little bubbles are hydrophilic. It means that they like water, and so they're oriented towards it. The tails that are all stuck in here towards the middle are called hydrophobic. Phobic, if I'm phobic of something, I have a fear of it, and so they're kind of afraid of water. And so everybody just kind of orients so that these tails are inside of the membrane and the heads are on the outside of the membrane, okay? It says here, but the DNA is still wound up tight inside of what's left of the cell. How do we unwind it, okay? So the first thing we're going to do is we're going to break down those membranes. We're going to use some soap to do that, but then the DNA is still wound up tight so that you can't see it. So I want to kind of show you what DNA is structured like inside of the cell. This diagram here kind of helps. Most people, if you see this, I noticed that on the RSU TV precursor before we come on, it even has like a little DNA strand on the side. This is your DNA. That's the double helix. It gets its name from the fact that double is the fact that it's two strands. Helix means that it has a twisted shape. As soon as you twist it, you've already made it smaller. Okay? Every time we twist it just a little bit more, instead of stretching it out, we make it smaller. And the whole goal here is that we want to twist it up as much as we can inside the cell to conserve space. All we're going to do today is undo all of that, okay? So then the next thing that happens is it takes this DNA and it wraps it around these proteins. In this diagram, they use little yellow circles for them. They're called histones. They typically occur in groups of 8, and they're going to wrap it around there, okay? It's coiled, it's twisted in the DNA double helix, and then it's wrapped around these histone proteins. And then we coil it some more, and we coil it some more, and we just keep twisting and twisting until it gets to this chromosome shape. The cell prefers to keep it this way, okay? When your cell is living its little cell life, for those of you that just studied mitosis and meiosis with Mr. Llewellyn, the DNA is all compact. We want to keep it nice and tight most of its life, but then it starts to spread out when we need to copy it. When it's all twisted up like that, it stays in that position until we need to use it. Your DNA actually codes for a bunch of different proteins. It says, okay, as this embryo is growing, make sure that it has 2 arms and 2 legs. Make sure that it has 5 fingers and 5 toes on each appendage. But it doesn't need all that information all the time, so some of them are turned off. When you turn on your DNA, it actually untwists so that it can be read, and then it retwists back again so that we can use it. Okay, so what we're going to do today to get that DNA out is we're going to do 2 different types of breakdown. When we studied this in physical science this year, we called it physical changes and chemical changes. So real quick examples of physical changes would be this ice cube melting and, unfortunately, the sad face of the shattered cell phone screen. What do these 2 things have in common? The thing that makes them a physical change is that they did not change chemical structure. I can take this water once this ice cube is melted and refreeze it back into an ice cube. I probably have a little issue here with this glass, but it's still glass, okay? Over here we have some chemical changes. We can have fire. When you set them on fire, then the chemistry changes. The oxygen is reacting with that. And then the other thing we have over here is baking soda with vinegar. It bubbles. Most of you probably have seen this with like the volcano examples that you can kind of create at home. The bubbles being given off is an example of a chemical reaction. Something has happened between the vinegar and the baking soda that allowed it to release those bubbles. Okay, so strawberry extraction. Here's what we're going to do. We're going to talk about the materials you might want to have at home. You can really do this at home if your heart desires. Strawberries. Found those at Homeland. They do still have them. Salt. You're going to need some sort of filter. I'm going to give you a couple different examples today when we go through the lab. I use an eyedropper for some of my lab. You can actually just use a spoon. It'll be completely fine. Little Ziploc bags. Usually have those on hand. This is the big money right here. Isopropyl alcohol is very difficult to find right now. My neighbor, Lauren, the one and only Lauren, little shout out to you. Thank you for the isopropyl alcohol. It has to be 91%, but everybody's kind of trying to get it right now to deal with the COVID issues, so that one might be a little bit more difficult. Might save this for later. I'm going to use a beaker in lab because that's what I do with my students, but you can use clear solo cups at home. Mr. Berg suggests using a mason jar. Guava juice, I always do this lab with guava juice. It's from Walmart. It's like about 50 cents, but you can use just water if you need to. Dish soap, you can use laundry soap, and then again, for the test tube, you can just use a clear cup. All right, here we go. So what we're going to do is the first thing you need to do is get you some strawberries, and I cut the tops off. You can't really use the leafy portion of the strawberry to do this, and you're going to stick it inside of a bag. I used 3 today. Typically, I'm only going to give my students 1 because they have a tendency to then try to eat them and consume them. But your job is close up the bag, try and get the air out, and then start squishing them, okay? When I squish these strawberries, which type of change am I going through? If I went back to those slides, is this the ice cube melting or is this the fire? Okay, is this a physical change or is this a chemical change? This is money answer, physical change. Okay, so what happens here is, yeah, I'm breaking this apart. These cell membranes are starting to break up a little bit. You'll notice, look how much more room it's taking up in this bag. Science vocab term hitting you right here, surface area. I'm increasing the surface area as well when I start to add these different chemicals so I can get some chemical changes occurring in here. I'm going to have more surface area for them to react, okay? A lot of times we'll talk about in zoology how organisms have these really long intestines. It's because they increase the surface area and lets them digest their food longer. In this case, we're going to be able to do more chemistry, more chemical changes with my strawberry. All right, so you mash this dude up. Try not to be too aggressive. Ziploc bags break. The Brody Wards of the world might destroy a Ziploc bag. Hi, Brody. But you won't because you're good. Okay. So this Ziploc bag. Now what we're going to do is open it up and start adding some things to make a chemical change occur. The first thing we want to add is 2 milliliters of soap. So my little eyedropper doesn't like the density of the soap. So what I'm going to tell you, and again, you probably don't have eyedroppers at home, is just maybe get like a spoonful. That's way too much. I was just kidding about that. Maybe like half a spoonful of soap. This is just Dawn dish soap. Stick it in the bag. Okay. What does Dawn dish soap do? Okay. If you've ever watched the commercials from old school Dawn dish soap, it is going to break down fat. Okay. Dawn takes the grease away. That's a fat. All right, so what we're going to do now is also add some salt to this bag. Again, it says we need to add 1 teaspoon of salt, and I just made my spoon filled with soap, so I'm going to use an old school little spatula, thank you, and add some salt in there. Zip the bag back up. Now, I'm going to mash it some more. Physical change, but now I've added the soap and the salt to start some chemical changes, okay? Think back to the diagram I had just a little bit ago of the cell and its membrane and little balloons, okay? Those phospholipids that are around the outside. Phospholipid is a fat. That fat, is that counting up or down? Sorry. This is, the soap is breaking down the fats that make up our cell membranes, and it is also breaking down the fats that make up the nuclear membranes inside of my strawberry. It's a plant, so it's a eukaryote, which means it has a cell membrane and a nuclear envelope. So we're just going to keep squishing around. We're going to give the salt and the soap the opportunity to chemically react with one another. All right, and open it one more time. And we're going to add some guava juice to it. Okay, the thing about guava juice is it has some enzymes in there that are going to also help to break down things, but enzymes also speed up reactions. Guava juice, this is just a little can I get at Walmart that's worth about 50 cents. And we're going to stick some guava juice in here. Now, not everybody has access to that. So, when we look at it, you can actually just stick water in here. If you look online, there's a couple different ways to do this lab. And it says if you add a half a cup of water to this, you're good to go. This is supposed to be about 4 milliliters of guava juice. So first of all, it's going to dilute it down. And
sorry,
no.
I'm going to stop. Can it be any type of juice? Can it be lemon juice or any type of?
I don't think so. I think it needs to be the guava juice. That is a great question. So it's going to increase your solubility here, okay? It's going to definitely make it less chunky. I mentioned earlier that we added that soap. I forgot to mention that when you squeeze this around, you want to try hard not to make bubbles in here, okay? It will be much better, and you're going to get some. It's soap. It's hard to avoid, okay? Make the bubbles for COVID-19 washing. Do not make the bubbles for the DNA extraction, okay? All right, so now what we need to do is start to separate this. When I break this down, it's still got some chunks in there. There's still some pulp in there. The seeds don't break down with this experiment, so we need to filter all that out. All we want to do is have the soluble portions of the strawberry pass through. So it tells you in the lab to use cheesecloth. I don't know how many of you have cheesecloth at home, but I'm not that girl. So what I do is I typically take a gauze pad from the cabinet, and if you open up a gauze pad, it looks an awful lot like cheesecloth with some cotton in there. And so you're just going to take that cotton piece that's inside and get rid of it, okay? But now there's a filter, okay? If you don't have any gauze pads at home, I've also seen this done with coffee filters. The whole idea is we want the solution to pass through, but we want to keep the solids out of it. So if you want, you just set that on top of your cup or your mason jar. And you might want to rubber band it to the top because this gets really thick. It gets a little bit more difficult to hold it. We're just going to pour this through. And I've had kids in the past, they're like, Miss Kehler, when I pour mine in, nothing passes through. I'm going to say maybe a lack of following instructions, but we can fix it, okay? Just add some water to it. It happens. It's all good, but we don't want the filter to separate through. All right, so now I'm going to hold mine because there's a lot of stuff in here. And what you're going to see is that there's a juice that's starting to run through there to the bottom. That's the good stuff, okay? That's the money right there. That's what gets you an A in science class this year. So I don't need a lot, but I need enough to probably fill up half my test tube. And again, what you're going to do at home, putting it in a cup and putting it in a mason jar, the more solution you have at the bottom, the more DNA you're going to extract. That's basically the difference here. So I'm going to squeeze mine just a little bit I'm going to squeeze mine a little bit. There we go. Yeah, I got a little nervous. That cheesecloth didn't seem like it was going to go. And there's a lot of soap in this today. All right. So that should be the money. Okay. And then you're going to try really hard not to make a mess in a fellow teacher's classroom. Okay, so there is my strawberry solution. We've gotten rid of the pulp, we've gotten rid of the seeds, and we got what we want. All right, so now what we're going to do is take a test tube. And again, you can just transfer this to a mason jar or a clear solo cup. One of the smaller ones would be better. You want to try and get the strawberry solution in the bottom, so try to just put it down one side. You don't want to like cover the whole inside. And I would say if you get about halfway up the test tube, that's probably good. I probably just messed that up for you guys all to see. And what you're going to notice is right away, the top portion is going to be a little bit lighter in color than the bottom portion. The soap is kind of rising to the top, but the good news is there's bubbles in there, and the bubbles actually start to carry certain materials to the top that we want to see. Right now, everything inside of there is dissolved. It's called soluble. We need to make it insoluble. We want a solid form of DNA to come out of this strawberry solution. Everything should be pretty soluble at this point. So set this down for a minute so I can get to the isopropyl alcohol. Thank you, Lauren. Isopropyl alcohol, 91%. What we're going to do is use this to create a viewing chamber. DNA is not soluble in isopropyl alcohol. And so when the interface of the isopropyl alcohol and the strawberry mixture meet, it starts to pull that DNA out. The bubbles from the soap allow it to rise. It's also less dense. That also allows it to rise. But the solubility is the main thing here. Because it's insoluble, it will start to make a solid structure for you to see. Okay, being very cautious. The deal with the isopropyl alcohol is we're also making what they call a viewing chamber. So from the top of the strawberry mixture to the top of this test tube, you want to have the isopropyl alcohol sit, but you don't want it to go down in here. We don't want to increase it that way. So very cautiously, I think it usually says in the lab we're going to add about 4 droppers full of isopropyl alcohol. You just want it on the top. So I tell my students to take the pipette, put it on the side, and just let it slowly run down. Oh, this is looking good. Okay. But I like my viewing chamber to be close to the top of the test tube. Oh my goodness, this is wonderful. And just because I love science so much, we're going to add one more. Okay, I lied. We're going to add 2 more. I'm just trying to get this closer to the top so that there's more of a viewing chamber for you to see. But you can already see this is starting to separate a little bit. You'll notice that it's significantly lighter. I should have brought like a dark background here. It's significantly lighter at the top than it is at the bottom. This is my strawberry solution that I put in to begin with. This is actually all the DNA starting to rise to the top. This is considerably more successful than when I practiced it the other day. This big white mixture at the top, it'll get better as the time goes on and it'll get bigger, but that's your DNA. I always tell students if it looks like you have snot at the top of your test tube, you're doing a great job. You did an amazing job. So I always tell them let it sit for about 5 minutes and then we'll come back to it and look at it. But you can already see there's red, there's clear right here because that's still my isopropyl alcohol interacting with the strawberry mixture, and it's just rising to the top. There's a big section of DNA at the top already. So I can pull this out. Oh my goodness, look at that. Okay, all of this white slime, that's your DNA, okay? And it'll just make more and more the longer it sits. I usually let them sit for a couple days just because the kids like to see them. Apparently it is also quite adhesive to a wooden dowel or bamboo rod. Okay, so I'm going to let that sit there for a minute, and we'll go back and kind of talk about what we were doing, okay? When we looked at our DNA lab, this is kind of what this is what it looks like at home. This is the test tube if you do it in lab class. So here's my strawberry mixture. Here's a little bit of DNA. We did way better than that today. And then this is what happens if you do it at home. This is a glass that somebody has at the house. All of this strawberry mixture, what I like about the glass that I didn't realize before I did this for you guys is that the viewing chamber is so much wider, which gives it so much more surface area for the interface of the strawberry mixture and the alcohol to react, which I think will give you more strawberry DNA. When I really, really like that. All right, so some questions to consider if I haven't had a heart attack yet, which I'm doing good. It says, why did we mash up the strawberry? Okay, what type of change was this? Was this physical or chemical? It was a physical change. Very good. So we just mashed it up by squishing it in the bag. It's still strawberry. I didn't change it chemically, so it's physical change. Okay. Number 2, why did we add the soap? Okay, the soap can be Dawn dish soap, it can be Tide laundry detergent. They're all going to try to do the same thing. They're going to try and remove the fat and break it down. The fat that goes around the cell membrane and the fat that goes around the nuclear envelope or around the DNA inside all needs to be destroyed. We're going to do that by using soap. All right, why did we add salt? We didn't talk about this too much. Salt helps to break down protein structures. There's a lot of proteins associated with DNA. Remember, there was 4 different types of macromolecules. There was carbohydrates that were sugars. There was fats, which we're breaking down with soap. There's proteins and nucleic acids. We don't want to break down the nucleic acids, but remember, they're wrapped around these histone proteins. And those histone proteins need to be broken down so that we can unravel it. That's what the salt's going to do for us, okay? That's what's going to break that down. And then finally, at the very end, we added this 91% isopropyl alcohol to create a viewing chamber. When I would ask students what it does for this lab, it's not just that it creates the viewing chamber, but it allows the DNA to become insoluble. It is dissolved when it's down here in this red portion, but it is not dissolved as it rises through that isopropyl alcohol. The bubbles help to bring it to the top. The fact that it's less dense helps it rise to the top. Sometimes you can get so much DNA out of your strawberry mixture that it actually just sits right here at the interface. It's so thick and so dense it doesn't actually rise to the top, but that is an amazing example. If you get that, you should take a picture of it and email it to me. That made me feel really cool as a great teacher. Okay, so if you liked it, awesome. If you didn't like it, email me and give me some ideas of what to do over the next couple weeks. Or if you have questions, you can do that too. Like, what did you say we were supposed to use again? I want to just take a minute to talk about the physical science activity this week. The physical science kids, for me, are studying Rube Goldberg machines. The Rube Goldberg is kind of like a giant game of mousetrap to me. I think it's fun. If this knocks this over and this knocks this over, and then you get to an end result. The original plan for Rube Goldberg machines, I'm sorry to tell you that we missed out on it, was you were going to build a Rube Goldberg machine that pied Mr. Berg in the face. He actually said yes. I was disappointed when I let some students know we were going to do that they asked, why not you? So I might have also gotten pied in the face. But I challenged you to build a Rube Goldberg machine as long as it had 8 steps to it and then it had some success at the end. You just told me what you wanted it to do. Yes, sir.
We can still do that if we need to on one of your lessons.
Yes! All right. It's going to be awesome. We can still pie Mr. Berg. Brett Burks built a Rube Goldberg machine that was trash dispensing. So at the end of it, it basically kicked a can into the trash can. And Tessa B was nice enough to share hers with you so that you can see it. If you listen to her, I'm not sure if you'll be able to hear her or not. At the beginning, she says, "This is take 14, Miss Kehler." So you have to be a little bit, competitive in order to get this to work. But this is her at-home Rube Goldberg machine she was kind enough to share with you guys. Maybe.
Today, attempt number 13 of the plant watering machine. Here it is.
Wow.
They were challenged to have at least 8 steps to it. I'm going to say say steps 1 through 13 trials were a little frustrating to Tessa, but I'm so proud of you for not giving up. Lindsay DeCamp emailed me one last night. Those of you that are having a hard time getting them to load to Google Classroom, all you need to do for me is on that third slide you're supposed to create, right? I emailed it to Miss Kehler, and then just email it to me. If that's easier, as long as I see it and you type it in your PowerPoint or your Google Slides, I probably won't forget, and then I can give you the 100 points that you deserve, because my son built one of these at home yesterday, and I really thought he was going to quit physical science. So I hope you guys have a great week. I will be back again next week. If you have any suggestions on what I need to do, you let me know. Thank you, guys.